complex that provided habitat diversity. You can see the extensive amount of channel, the extensive amount of water interface with these emergent tules and other emergent vegetation that provided high productivity within the system. It also provided areas of habitat for foraging and for other functions for those fish and macroinvertebrates that inhabited the Bay Delta estuary. And it was partly as a result of this habitat complexity that many of the habitat requirements for various species, such as low velocity areas for juvenile rearing, were met over this wide landscape level of changes that occurred historically within the system. As a result of the changes that have occurred within the physical habitat of the system, there are a number of biological functions that flow historically supported and provided that are either diminished or no longer available. They include the loss of access to wetland habitat that was important for juvenile rearing and foraging as well as food production, the loss of access to seasonally inundated floodplains that are now largely managed for flood control purposes and only incidentally provide fishery habitat, the loss of these shallow water channel margin habitats that were important for juvenile rearing as well as migration, and all of these have contributed to reduced production of organic material and food supplies within the system. And we've heard a lot of discussion from the State Board, from the Bay Institute and others about the unimpaired flow construct and how that might help restore some of these various functions. And it's important to recognize that although increases in, say, winter and spring flow may have benefits, although uncertain for some species, there's also a potential risk of adverse consequences for other species. And I'd like to just briefly describe that. These are results of hydraulic modeling that were produced by Walter Beret from MBK. They were submitted to the State Board in 2012. And what they show are results of reservoir storage for the Trinity River, the Sacramento at Shasta, the Feather River at Oroville, and the American River at Folsom. The red line is the result of the simulation model for reservoir storage under current operations. The blue line is the simulated reservoir storage under a 50% unimpaired flow regime. And I'm not presenting these results in substance, but rather to illustrate a couple of important points. One is that as a result of the fact that these salmonids are now spawning lower in the system, for example, winter run spawn in the area from Keswick Dam immediately downstream. They spawn during the summer months when air temperatures in the Redding area are frequently in excess of 100 degrees. Water temperature has been identified as a critical factor in terms of the survival of incubating winter run eggs. And the ability to maintain cold water in the area where these eggs are incubating is largely a function of reservoir storage and cold water pool management. So as we start looking at various alternatives, it's important to look at what effects those alternatives would have on processes such as cold water pool and reservoir storage. These results then would typically be linked to water temperature modeling. The water temperature modeling then provides the input to egg mortality modeling, to juvenile rearing habitat quality in a model such as SalMod, and ultimately into a life cycle model such as IOS to really evaluate the various pros and cons of a given alternative strategy and how it might adversely or beneficially affect various species. And this just summarizes the points I just made. In terms of the functions that flow provides in terms of physical habitat within a river, this is a depiction of an unaltered river system. You can see there's a low flow channel towards the center of this graphic. This is slide number 47. There's emergent vegetation along the channel margins. And as flows increase, the water then comes out over the sides of the channel and inundates these floodplains. That inundation of the floodplain then provides additional mechanisms for juvenile rearing as well as the transport of organics and foods further downstream. But it also interacts with the riparian vegetation that grows along both sides of the channel. And that's how we'd like the system to look and how it should function. This is more typical of the Sacramento River. Channelized trapezoidal channel with riprap sides. And you can see in this example that a change of flow actually doesn't produce very much in the way of a change in habitat quality or availability because of the physical changes that have occurred within the system. And so those are important elements to consider as we move forward in looking at these various flow paradigms. So how are we addressing this issue? In the context of the current thinking, and it's reflected in the BDCP planning effort, a big element of reestablishing these flow habitat functions is through restoration activities. BDCP proposes to restore 65,000 acres of tidal wetlands that would then provide this additional rearing habitat, foraging areas, and cover. Changes to the frequency and duration of access that fish would have to floodplain areas like the Yolo bypass is being addressed through changes like notching the Fremont Weir so that those floodplains then function at a lower flow and provide greater opportunities for access. Shallow water, low velocity habitat along the channel margins can be achieved either by resetting the levees further back or by creating additional benches that can then be inundated and provide the habitat functions that the trapezoidal channels no longer take. We've talked a lot about uncertainty, and a number of parties have legitimately raised concerns about habitat restoration and reestablishing this flow functional habitat interaction. And the way we're addressing that is largely through interdisciplinary monitoring in habitats like Liberty Island that we heard yesterday are functioning well for many of the fish species, including Delta smelt. Looking at the result of the monitoring in those locations, identifying the metrics and the attributes of those types of habitats that are functioning well, in contrast to other habitats like Frank's Track or Mildred Island that are not functioning as well. And also through collaboration with investigators in the Pacific Northwest who are already implementing these kinds of habitat restoration projects, and they're monitoring and learning, and we can take advantage of that. Some of the things that we're learning is that the habitat design should really reflect the habitat suitability requirements for a variety of species, and that it should be designed in a way that it functions in terms of the natural processes, the tidal dynamics, the sedimentation, the circulation patterns. Those types of processes need to be integrated into our habitat designs to reduce our uncertainty. We're developing better models, we're developing better biological and water quality and other information to help make those decisions. We've recognized the importance of providing habitat complexity in terms of bathymetry, tidal currents, emergent vegetation, other elements that help improve that functional performance of these restored habitats at various flows. And we're finding that by dispersing these habitats geographically throughout the region of the Delta, we can provide benefits over a range of flow conditions to a wide variety of species. And it's important to recognize that multidisciplinary collaborative monitoring is going to be an integral part of reducing uncertainty. And as we design these monitoring programs, as we design the habitats, we need to embrace the adaptive management concepts that you've talked about, but we need to do that up front, not as an after fact, so that we can take advantage of the monitoring, and we better structure the monitoring to provide those answers. And the last thing I want to touch upon is just a recent study that was released by Winder in 2011. it looked at the various factors that contribute to invasive species colonization within the delta. And there's no question, I think there's broad consensus that exotic non-native species have changed the ecology of the delta, the trophic dynamics. But Winder actually cited that long-term droughts and increased salinity intrusion were two of the dominant factors that allowed for greater expansion of these non-native species. And in their analysis, they simply said, based on those kinds of conditions and the fact that water exports have been increasing over time, water exports then have exacerbated this colonization process. And what they didn't do is the kind of more integrated analysis that looks not just at a simple assumption that exports change salinity intrusion, but rather at the types of information that Paul Hutton was showing, where you really need to look at upstream reservoir operations, releases during drier periods to maintain the D1641 water quality criteria. It's a much more dynamic and complex set of interactions that they didn't take into account in their paper. And so as a result of that, I don't think there's been any kind of a rigorous analysis of the role of water exports on invasive species in the delta. And rather than being characterized as a fact, I think that should be characterized as an untested hypothesis. And with that, I'll turn it over to Pat.
Thanks, Chuck. Our final speaker will be Dr. Pat Glibbert, who will talk about nutrients in the food web.
Thank you, Dave, and also thank you for establishing my relative age on this panel earlier. Good morning, Madam Vice Chair and panel. Thank you for the opportunity to be here. I want to talk about nutrients and the food web, and while it may seem that this is a singular stressor talk, I hope you'll see that this is actually a multiple stressor approach that I'm taking. The first thing I want to talk about is the issue of ammonium, because it's been an issue that's been discussed at great length in this system, and there's a great deal of controversy or uncertainty regarding the role of ammonium. And the way I like to look at ammonium is as a paradoxical nutrient. It has yin-yang properties. On the one hand, it can be a preferred nutrient form of nitrogen for phytoplankton under some circumstances, but under other circumstances, it can be inhibitory or even toxic. But it's well known that sensitivity to ammonium may be a universal biological phenomenon. In fact, I want to underscore that there's absolutely no controversy or uncertainty regarding the effects of ammonium on plants, including algae, and animals, including humans. That is, ammonium metabolically behaves differently than other forms of nitrogen in plants and animals. The threshold at which those symptoms of preference or toxicity get manifested certainly differ, and they differ depending on the species, the conditions under which the ammonium is supplied, the physiological state of the organism. So it's certainly understandable that there are differences in the literature with regard to this threshold. The threshold that's been reported for algae in the literature ranges from 1 micromole to greater than 20, so a 20-fold range. In this system, the number that's been often used is what's called the Dugdale threshold of 4 micromoles. Sometimes you see it in the units of milligrams per liter. But this term 4 micromole has become operationally appropriate in this system. And you can see ammonium has increased in the Bay Delta over time. And this Dugdale threshold was crossed right around 1990 or so. And the reason we call this a threshold is that at concentrations above that threshold, chlorophyll does not accumulate at significant levels in the water column, whereas when ammonium is lower in concentration, those chlorophyll levels can accumulate. And this kind of relationship has been seen worldwide. The inhibition of ammonium by diatoms is increasingly recognized in the global literature And in fact, it's being included in ecosystem models of global ocean productivity. The bars that you see there are the number of citations in the literature to ammonium inhibition of diatoms. And you can see just in the past decade or so, it's really become a universally incorporated phenomenon in our understanding of phytoplankton physiology and global productivity. But our singular focus on ammonium has blinded us to a number of other changes in nutrients. And I want to look at another nutrient that has also changed, and this is phosphorus. Shown here in the graph is the total phosphorus load from the Sacramento Regional Wastewater Treatment Plant, and you can see that threefold drop right around the mid-1990s. And that was not due to an engineering change, but rather due to the introduction of phosphate-free detergents. And those detergents were introduced to basically the entire developed world right around the same time. So what has happened overall is ammonium has increased, phosphate increased from the 1970s to about the 1990s, and then dropped off. As a consequence, the nitrogen to phosphorus ratio increased in a near linear fashion over time. Now, why are we interested in these nutrient ratios? Well, we classically think of nutrient ratios as helping us to define the nutrient that is limiting production. So the growth of phytoplankton is limited by the nutrient that is in least supply relative to the needs of the organism. Now we have somewhat of a conundrum in this system though, because even though phosphorus has declined, it hasn't declined to levels that are vanishingly low. And when we take a simple classic metric such as half saturation constants, it doesn't seem to be limiting. So even though we've changed these proportions, we haven't really driven the system into what would classically be considered a phosphorus limited system. And the classic dogma says that as long as there's enough of the resource around, the relative proportion of those nutrients shouldn't really have any regulatory influence on defining the species composition of the community. That's the classic dogma. Now that classic dogma has actually been replaced in the last 10 years or so by some new thinking that says while the total nutrient load sets the absolute amount of productivity or biomass that a system can support, it's the relative proportions of those nutrients, whether they're limiting or not, that sets the quality, who's there and how they're doing. So it becomes a study of the balance of chemical resources and their interactions, and it focuses on the match or mismatch between the differential requirements by different organisms for nutrients and their availability. Now this perspective has been formalized with the term ecological stoichiometry or ecological ratios, and there have been books published on this, but it's really been in the last decade that this thinking has evolved. And here you can see the numbers of papers published each year on ecological stoichiometry. And it's really just been in the past decade. So now 60 papers a year or so are being published on this topic, not just in the aquatic sciences, in the terrestrial realm, and the concepts are even being used to understand, for example, the regulation of the growth of cancer cells relative to non-cancerous cells. So stoichiometric modeling is beginning to transform the theory of food web ecology. So what does this all mean? Basically, the idea is that it's a grand balancing act. Organisms take up and use what they need, and they have to get rid of the rest. Too much can present metabolic costs, as does too little. And different organisms have different needs for different types of nutrients, and they have different strategies to regulate their biomass. So it takes a different kind of nutrient proportion to make a clam, for example, than it does to make a striped bass. So the idea is that as we change nutrient loads, forms, and ratios, we first change cellular metabolism. That can be expressed as a change in growth rate. When we change growth rate, we can ultimately change the community composition because other species that have other mechanisms can change and become dominant, and ultimately that can drive changes in the food web. And these changes are fundamentally predictable based on the biological demands of organisms. Now, if we start at the base of the food web, the algae, we think of them as following essentially a you are what you eat model. They don't have a lot of capability to regulate their nutrient content. They have some, but in the grand scheme of organisms, they have relatively little. But there's a huge diversity of algae in the microbial world. And I love this figure, which was taken from the literature. But what it shows is that across species, there's a huge diversity in size, composition, and in growth rate. So just shown on the top are a number of representative type of algae that you might see in a microbial sample. And at the bottom, it illustrates that the range in size of those species can be as large as comparing a fish to the city of Manhattan, even within a microbial world. And so those species obviously range in cell size, but they also range in their cellular composition of different elements. So their carbon to phosphorus ratio, for example, changes tremendously. Not advancing. So as we change the nutrient forms or ratios, we first can change the cellular physiology within the ability of those species to adapt to that nutrient change. There's a limited ability to change cell size. They may change their elemental composition, but mostly their metabolism changes, their enzyme activities, their cellular pigmentation, which is their ability to carry out the process of photosynthesis, ultimately growth rate changes. And one of the things that we're learning more and more about is that at least in some species, an imbalance in nutrient ratios can lead to toxin production. So even good food can go bad. And you've heard about the fact that many copepods, for example, don't eat diatoms. Well, diatoms really are a significant source of food, but they too can go bad when the nutrient resources available for them lead to an imbalance in their nutrition. So what does this mean with respect to the kind of algal community compositional changes that we've seen over time? Shown here is the change in diatoms in the Bay Delta and the change in the nitrogen to phosphorus ratio over time. And as we heard earlier, that nitrogen to phosphorus ratio also incorporates that increasing ammonium content. And what you see is a near perfect mirror image relationship. As the N/P increased, diatoms decreased. But while you've heard about the decrease in overall chlorophyll content and concentration in the water column, and now this decrease in diatoms, there are actually some species that are doing very well. The cryptophytes, dinoflagellates, and the cyanobacteria, as you see highlighted in the circles, have increased over time. Some of these cyanobacteria are the toxic cyanobacteria. So it's not just that the total quantity has changed, but the quality of that algae has changed as well. So as we change the quality of the algae, what effect does that have on the next level in the trophic web. What effect does that have on the grazers? Metabolism would suggest that indeed it would have an effect. And metabolism begins with diet. As we change diet, changes in diet can lead to changes in reproduction, which can lead to changes in egg viability, ultimately growth rates, and population success. So as we move from phosphorus-rich to a phosphorus-poor diet, for example, it can change metabolism. Now, the principles of ecological stoichiometry tell us that nutrient ratios have effects at every stage of the food web. But an animal's elemental composition is linked to its evolved structure and its life history. So it's going to have different needs for nutrients as larvae than as adults. And it takes different proportion of nutrients to make different types of structures within organisms. So we can fundamentally build different organisms just knowing their proportion of skeleton to muscle, for example. And therefore, different types of organisms should thrive as the nutrition changes. But at higher trophic levels, the secondary consumers, the grazers, are finely tuned with their prey, but they don't follow this you are what you eat model. They follow more you are what you the opposite. They graze at opposite ends of the spectrum often because they have mechanisms to regulate that which they don't need. They can excrete that which they don't need. They can keep what they need and excrete that which they don't. And this keeps this balance by providing those nutrients ultimately to the primary producers again. And so when we look at the changes in time in this Bay Delta system, we see that as the diatoms declined, this is looking at annual average data, as the diatoms declined, so too did the grazer Eurythemera. Very tightly coupled relationship. And it's more than a correlation. Here we know that this is driven by the mechanism of grazing. Now you saw this earlier. This is the shift in zooplankton community composition. The Eurythemera copepods dominated in the 1980s, whereas currently the community composition of zooplankton is dominated by this very small single chicken nugget Limnothoina. There really were a whole sequence of other species that filled in that gap. It wasn't as if the system went empty in the 1990s, but we're just showing the two extremes. These two organisms actually have a very different elemental composition of their biomass. And when we ratio the two, the abundance of the two, that ratio actually, again, is a mirror image of the N/P ratio in the water column. And you can see that correlation again. So that correlation, again, is driven by a mechanism driven by nutrition that would be predicted by the elemental needs of these two species. Now, we can carry this a bit up the food web. This is the relationship between longfin and N/P. And again, you can see that there is some suggestion of a mirror image relationship. That is, as N/P rose, longfin declined. Again, keep in mind that this increase in N/P is a function of both the increasing ammonium concentration and declining phosphorus concentration. Now, you've heard about the rise of the centrarchids. This is quite interesting because the centrarchids actually track the N/P ratio not in opposite mirror image form, but actually in parallel. And it has been stated that as one ascends the food web, trophic groups grow increasingly nutrient and especially phosphorus rich. The centrarchids are especially phosphorus rich, And you see that that's been documented in the literature. They have more phosphorus content in their biomass than do cyprinids, for example. And this is because they have more bony structure. But again, that puts them in a position where they are becoming more prevalent when there's less phosphorus in the water. But that's because they have highly evolved excretion mechanisms to help them balance their nutrient stoichiometry. And so again, just like we saw with the copepods and the phytoplankton, they don't have to eat the same elemental composition as their biomass. So the kinds of nutrient changes that we've seen are that we've seen increasing ammonium, and we've seen increasing nitrogen to phosphorus. And what that has resulted in is an increasing dominance of organisms that are either ammonium tolerant or can make do with fairly low amounts of phosphorus, as some of the cyanobacteria can, or can sequester the phosphorus very efficiently and excrete the excess nitrogen, thereby accelerating that increase in nitrogen to phosphorus in the water column. The phosphorus gets bound up in the biomass and is not left over in the water column. So over time, what this has meant is that no matter how we examine these data, there are winners and losers as a function of this change in ammonium and phosphate. Some of the losers are overall chlorophyll, diatoms, we saw Eurythemera, neomyces, and longfin. Some of the winners are dinoflagellates and some of the other cyanobacteria, the other types of phytoplankton, limnothoina, and the largemouth bass and sunfish. Now, some of these winners and losers, using the principles of ecological stoichiometry, are fundamentally predictable. Some of the winners actually are invasive species. And I want to spend a minute or two talking about two of these invasive species, the clam, corbula or Potomac corbula, goes by both names, and the invasive weeds that we've heard about, the Aegeria densa. And I want to make the argument that the invasive species, or some of the invasive species, may be as much a response to nutrient and ecosystem change as they are a cause of ecosystem change. In other words, they can thrive in these altered ecosystems, but would not have been able to thrive in the more natural system. So two of the invasives that have very much structured this system are the clam, which we've heard about, its enormous grazing capability, and the invasive weed Aegeria densa, which also provides habitat for the large predator fish. Now, if in fact there is a relationship with entropy, we would expect to see that this same trajectory might be seen in some other systems. This is the Rhine River, and the blue here shows the cumulative number of invasive species over time. So this too is a heavily invaded system. The red is the change in nitrogen to phosphorus ratio with time, and you can see here, as indicated by the arrow, this too was a system invaded by corbicula, a related clam, and Aegeria densa. Now, you've probably heard about the Potomac River and the fact that it was in the 1980s successfully restored by the removal of phosphate from the wastewater treatment plant. And shown here is the change in the white squares is the N/P ratio of all the loads in the system, and you can see it increased very rapidly from the 1970s to the 2000s. The red shows the numbers of acres covered by submerged aquatic vegetation. So on the one hand, this has been touted as a success story. The river was successfully recovered in terms of the water weeds. On the other hand, these are invasive water weeds. This is hydrilla, which is very similar to Agariodensa. Similarly, in the Potomac, as N2P, shown here in two ways, both as all loads and as just in the effluent of the Blue Plains sewage treatment plant. Corbicula began to appear when the N2P first rose. It reached a maximum density when the N2P was at its highest, and corbicula began to decline when nitrogen was removed from the sewage effluent. Here is another example. This is the lower Ebro River, Spain. We only have phosphate data here for this system, but you can see in 1995, phosphate declined significantly, and it was at that point that the invaders, hydrilla and corbicula, began to thrive and do exceptionally well. And my last example here is from the lower Hawkesbury Nepean River in Australia. In the mid-1980s, you can see phosphate shown in the pink dots was very high in concentration, and Agariodensa wasn't present at all. Phosphate declined in the mid-1980s. Agariodensa now covers much of the length of the river. Corbicula has increased, and in fact, toxic cyanobacteria have also increased as well. This system is also one driven by exceptionally high nitrogen loads from a sewage treatment plant. So I'd like to make the case that ultimately these stressors are all connected. So rather than thinking about them as multiple independent stressors, they really are connected. Nutrient loads set the overall production of the system. The proportion can set the quality of the food web, who's there and how they're doing, and ultimately then how they interact also controls the relative proportion of which organisms are there at any given time. The proportion of nutrients can also provide opportunities for invasive species to thrive. Whether those invasive species were present in low abundance earlier or not, it's the change in proportion that allows them to do well, and they too then can restructure the system. And those nutrient loads and proportions are maintained in balance not just by what's coming into the system, but the internal recycling, both by the organisms and as well by the sediment chemistry. And we need to keep in mind that the sediments are in fact huge reservoirs of nutrients, and the sediments really are the history of the nutrients that have been loaded into the system for many years. Phosphorus particularly remains in the system for a long time. And the fluxes, the exchange of the sediment and the water column in terms of nutrients can change with salinity. It can change with pH, which can change with the productivity of the system, and of course with temperature, and there are many complex interactions that are regulating these interactions. But phosphate fluxes tend to be higher in salt water, whereas ammonium fluxes tend to be higher in fresh water. So the way I like to think of nutrients and flow is this, again, back to this yin-yang relationship. The residence time can be thought of as an exposure time of organisms to either beneficial nutrients or to those that are inhibitory or toxic. And it depends on how long the biological processes have to react to those substances. Flow can bring in new nutrients. It can dilute point source nutrients. It can alter the reactions at the sediment surface. And it can export nutrients downstream where it can contribute to blooms displaced in time and space relative to the source. So it is now well established that many offshore blooms around the world are related to sources of nutrient far upstream. So we can think of the whole system as being in dynamic balance, and I think nutrients are a significant part of that balance. And that balance has to consider the yin and yang of preference and inhibition, the metabolic costs of too little and too much, ammonium and nitrate, nitrogen and phosphorus, grazing control versus nutrient control. And food webs are ultimately shaped by the balance between nutrient resources and the elemental demands of the organisms that are present. So just to recap, ammonium is a paradoxical nutrient, and so it's understandable that there's some controversy or uncertainty regarding its effects in the ecosystem. Nutrient ratios do matter, altering the quality of the food at all levels, while nutrient loads set the absolute amount. Too much can be a metabolic stress, as can too little. And the Bay Delta is not unique in the trajectory of many of the food web changes that have occurred with changes in nutrient loads and ratios. These nutrient changes can alter the ecosystem, providing opportunities for invasive species to thrive when they would not otherwise. And this concept of ecological stoichiometry can help us by at least providing one mechanism whereby we can begin to understand these relationships between fish and flow. And I'll end with a quote from one of the pioneers of this concept of ecological stoichiometry. That is, consumers can be destined for extinction when faced with poor food quality. Thank you.
Thanks, Dr. Glibbert. If you could advance that one slide, I think I have kind of a wrap-up slide. But I'll do it very quickly because I want to have some time for questions. Okay, I'm not going to worry about it. The bottom line point that I would like to leave you with is a paraphrase from my real estate agent, which is mechanism, mechanism, mechanism. The goal of science in order to serve the needs of the board I think should be to tease out the mechanisms. What causes what? And of the mechanisms, what can we change? I mean, temperature may be an important mechanism, but we can't do anything about it. Other things may be things we can do something about, but may not make much difference. The goal here is to find mechanisms that we can invest in or regulate and actually make a measurable difference to the target species. Based on our review of the literature, at this point, we think that of all those various interventions that are possible, that habitat restoration and nutrient regulation offer the most promise. But obviously, this is a story that will continue to evolve as the science evolves. So with that, I think we're done.
Thank you, panelists, for all the presentations. It's a very rich amount of information. I think it gives us a new challenge in terms of balancing, right, Dr. Glibbert? Because balancing nitrogen and phosphorus as well as all the other balancing. I found it very interesting with my background in biology and wastewater engineering, where we look at stoichiometry in terms of some of the fundamental equations behind wastewater engineering. So conceptually, it's not a big stretch for me. One of the questions I had from yesterday, actually, so I can kind of bring it back, I have also a lot of interest in the invasive species issues. You may not know this, but I was on the Federal Aquatic Nuisance Task Force for 4 years. So I got to go on field trips to a lot of places where we had invasive species around the country. And I'm continue to be active and interested in ballast water treatment and the engineering and the development of that, preventive aspect. So the Egeria question and, its ecosystem change causes, but also, talking about how it gets established. And so, and panelists can speak to this, also the science panel too. I wasn't seeing, with the flash studies this year, did we see a suppression of Egeria? Because that might be a longer time scale response. We saw a lot of effects, that were anecdotal, I would call them at this point. But I found it compelling, this idea of the nutrients and, encouraging the growth of this particular invasive. And so my question to Dr. Glibbert is, in the case studies around the world where nitrogen has been controlled, with one of those dials we can turn, control, right? It's the C in SWRCB. That's what we're interested in. What can we control? Where it's been controlled, have we seen a response in terms of Egeria densities and then commensurate, centrarchid type response?
In terms of the responses around the world, one of the most significant studies is from the Potomac. And Egeria has, or its equivalent, which is hydrilla. They're very similar plants. So we have hydrilla. The abundance of hydrilla has declined with the removal of nitrogen from wastewater treatment plants, and we've begun to see the emergence of more native species. So that is one that we've begun to see a reversal. It does take time. It is a very aggressive invader, there's no question. And we know that it invades for a number of reasons, but it thrives when the environment is right. And it's a fascinating plant because it is ammonium tolerant. It tends to grow, for example, in fish aquaria. So we know that it's ammonium tolerant. It is able to use the sediment resources for its nutrition as well as the water column for its nutrition. And it grows very productively. When it grows rapidly, it can actually raise the pH in the water significantly. So productive Egeria or hydrilla bed, by the end of the day, can be up to several pH units higher in pH than normal seawater. The consequence of that is twofold. One is it's actually been suggested in the literature that it too grows tenfold faster at higher pH than it does at lower pH. So it's a self-reinforcing mechanism. So it can grow under its own self-induced toxic conditions. The other mechanism that then itself helps to promote is by raising the pH, phosphate begins to efflux from the sediment. So it changes the nutrient dynamic significantly just by its very growth, because it sets in motion all of these biogeochemical processes that then other species can take advantage of.
Yeah, that was very interesting. Thank you. And it's nice to actually see you in person, having read your stuff. So you're the rock star, sort of the. No disrespect intended. I'll now go read everything else you guys have written, but it was not disappointed. So very interesting. One quick question without diving into any of them or figuring it out. I'm assuming, Dave, since you are going to come back to multiple panels, that at some point in this series of presentations, you're going to suggest what mix of stressors make sense. I get your point today, which is it's not just flow, which is what you're trying to point out, and some suggestions of what the other ones are. I appreciated hearing that. But are you going to be proposing something to us by the end of this process?
Yes. Yeah, so I mean, we're viewing this as it's an extended process, a number of different workshops. So we're going to try to unveil things as we go along, but we're still working on some, our list, but and our review of the literature. That's our intent. Yes.
Okay. Because interesting thing in terms of I get your point about correlation. It's not just correlation, it's also causation and mechanisms, but there are all kinds of instances in which we can't know the precise causation before we need to act. Smoking being the key one, where from a public health standpoint, we didn't have the causation mechanism down until very, very recently, but we knew enough to know there was an impact. On the other hand, I know correlation alone may not be enough, but I would just encourage you to be more holistic in that, because I know one man's correlation is another one's creed, but it's still we need help figuring out how to do the mix of these. And
Right.
the answer can't be it's not flow at all.
Right. That isn't our answer. We will be presenting a lot more information. I think that will be of interest to you as we get into the pelagic workshop, including results from life cycle models and so on that start to get at some of these questions. And I'd be happy to also to show you other correlations that are just as strong as the flow correlations. This whole system is kind of cross-correlated. This is part of our problem, is that you can find a flow correlation for fish, you can find an ammonium correlation for longfin, you can find a turbidity correlation. I liken it to a hall of mirrors. You're in here and you're seeing all these images that are approximately as good as each other. And what you're trying to do is figure out which image actually is the real one and which is just a cross correlation with another one. That's, to me, the challenge here.
That's it for now.
Yeah.
He has another one.
I have one question for Richard or Diane. In terms of cold water pools, haven't we been getting some modeling done on various flow regimes and the effect on cold water pools?
Well, when we have a suite of alternatives, we'll certainly analyze that effect, but I would point out that the results that Dr. Hansen provided, I think, were assuming that none of the flow was taken from consumptive use diversions, and you're seeing those reductions in cold water pool storage for that reason. So if you were to reduce diversions, then you could, of course, maintain some of that storage.
Good. Yeah, and building on both of your comments, fellow board members, I mean, that was sort of what came out of the presentation was, don't touch that, it's working now. Whereas we've, to meet the existing Bay Delta plan, we've had reservoir releases, and there's clearly been benefit. Maybe there's been some cost, but I think that was an incomplete story that we received about, the effects of reservoir releases on Delta resources. I'd like a little more of the whole story.
I think what maybe it indicates is that we need to look at carryover storage requirements in our planning process too, and would like to receive information from the fish agencies and other interested parties regarding what kind of criteria we should be considering in that regard also.
And a question to Patricia. Once you mentioned in some of the review of the different international examples that you were giving, you mentioned denitrification, and then the numbers went up for some of the species that before that had been going down. And I'm assuming that in the Chesapeake they with their large treatment plant they did denitrification. Is that one of the levers that you see to help create some of the balance?
I think the Potomac River is a really excellent model system to look at in terms of this system, because the Blue Plains treatment plant, one of the largest, undertook phosphate removal in the 70s, but then a series of steps of nitrogen removal in the 80s. And I'm not going to get these dates right off the top of my head, but the first step in nitrogen removal removed about half of the nitrogen, and then we're down to now about 3 milligrams per liter in the effluent, and the discussion is to reduce that even further. But yes, we've certainly seen ecosystem changes with each of those step changes in nitrogen.
And I'm just going to kind of piggyback a bit on what Felicia just said, and I thought the analogy with smoking is an excellent one, because you all know that we are going to be making decisions before we know exactly what's what, and that we may not know what's what ever. Because it's always changing, and so that's just the way things are. And so, what are those things that, while we may not know the causation, we may not know the why, but we do see the effect. We do see some positive effects, and what are those over which we have some influence, and over those that we can encourage influence in other areas. And so it really isn't going to be helpful to me for you to talk about unimpaired flow versus natural. I get that. Or that flow, and as you say, you're not saying flow is not the problem, but it's but it is associated with many of the things. It's associated with the food web. It's associated with some of with habitat. You were talking about plains inundation. You don't inundate without water, and so it's associated with all the temperature. It's associated with turbidity. It's associated with all these things, and so we're going to need. One of the things that I like about the materials that you have presented to us, which I really think are quite good, is that you're saying, let's think about this in the context of of nutrition, of these various other factors. Let's think about flow in those ways, not just as a thing unto itself. And I like that. That's absolutely what we should be doing, and we should be doing a better job of it, because we've always actually been there, but we just have done, I think, a very poor job of demonstrating that we're thinking in those terms. So what we will need, I think, from you as we move through these workshops is help along those ways, is not that flow. How does flow relate to these other factors in your minds? And how do we take that into account?
We can definitely address that, and we intend to address that in our next round that you'll be getting.
Okay, so everyone's held up really well. It's now time for our lunch break, so should we
1:30.
come back at 1:30?
Mhm.
Okay, we'll start again at 1:30.
Okay. Got to get my shoes on.